house today.
Oh, really?
And I'm like, I know that. I know that.
Really?
Once you
What did he say?
Yeah, I think that goes for everything.
I think he was generous.
Not necessarily
That was the
business,
best
is it?
test.
You want to see
Chemistry
that?
actually tells them how to play.
What does that mean? You only learn it if you don't.
I mean, if this gets I get out, it's fine.
Go early if you
I
want.
think just because we're in the same room, like, for example, like, I have like resistance problems.
And I get him started.
So good, I like my college now.
Just the 30 minutes
Just
for
sitting.
one.
Like I know how to do occasionally in the bar.
We'll start when you're ready, guys.
Oh, man. Probably succeed. More.
Full body regulation in here.
Like when I tutor people, I'm kind of like, how do you not know this? Because it's like sad that you recognize it, but like after we're like, oh,
Do you ever tell them that?
like what?
Do you tell them how you know?
No, I don't say, why do you not know this?
Just think of that.
That's really rude. I don't want to.
Destructive.
No, there's a good story.
Yeah.
And that's how I grew up.
Separate yourself from the world.
Okay, let's go. Thank you guys who are here. It didn't really occur to me that you'd be the last people taking a class, the last afternoon. But thanks for being here and for getting your exams in. Any issues or questions? I think I got one from everybody. So, as I said last week, we'll be halfway done today and pretty much done with all the heavy lifting. So you've just about seen every part of the immune system so far. So the second half of the semester will all be review of things we've learned already, but more about things that go wrong, why we end up with infections, inflammatory diseases, etc. So we'll round off T-cell immunology today with development and activation, and have a discussion in between. Anybody have? Okay. So we talked about a few classes ago that B cells develop in the bone marrow, and they are not fully competent to have immune responses until they've been to the spleen or some other lymphoid tissue. And T cells are similar in that they come out of the bone marrow, not capable of an immune response, but go through education and maturation in the thymus. So that's a lot of what we'll spend the first part of class on. And then they recirculate throughout the body, and they keep moving for the most part until they've gone through an immune reaction where they may develop a preference for one particular part of the body. So the thymus is not just immune cells, but has some very specialized epithelial cells. The embryonic tissue from which they originate is not that important, but it's just, it has structural cells, it has specialized epithelial cells we'll talk about later, and then it has a number of antigen-presenting cells, as well as the thymocytes themselves. So there's hematopoietic cells and structural cells. And it's populated pretty early, day 12. During development, hematopoietic cells start to enter the thymus by migrating out of the circulatory system and then differentiating within the thymus into antigen-presenting cells and other cell types that populate the thymus. And then later, the T cells come from the bone marrow. Okay. Okay, so we'll break it into much more detail, but I just want to integrate what we talked about last time in terms of TCR gene rearrangement and when that happens. So we'll look at this in a number of different ways, but the immature lymphoid progenitors, which have no T cell receptor and have no CD8 or CD4, so they are hard to identify as a T cell at all at this point when they enter the thymus. They undergo DJ joining followed by VDJ joining on the beta chain and reach a checkpoint. And at that point, once they've done a successful rearrangement of the beta chain, they start expressing CD4 and CD8. And during this time, they're increasing CD3, which is a signaling component of the T cell receptor as well. So basically, just like the different stages of B cell development, the T cell receptors are undergoing continual changes in their cell surface receptor phenotype. And then they have checkpoints, which we'll go back over, checkpoints for successful T cell receptor rearrangement. And if they don't successfully rearrange their T cell receptor, they don't survive, just like B cells don't survive if they aren't successfully rearranging the BCR genes. And so the end of all this process that's occurring in the thymus is the cells becoming single positive. So they don't decide to be a helper T cell or a cytotoxic T cell until after they've reorganized their TCR and gone through negative and positive selection. And then, so it's very late in the process. They pick one or the other. So I know it's complicated, but is that relatively clear? Hi. It's okay, unzip. Okay. So those different stages, they're continuous with changes in gene and cell surface receptor expression, but they can be divided into named stages. So there's a common natural killer and T cell precursor that goes through 3 stages during the double negative stage, which is describing these changes in cell surface marker expression. At the end of those 3 stages, they have either successfully organized a gamma delta chain. If they haven't, then they start organizing an alpha beta chain. And your book, most of what we're going to talk about is alpha beta T cell rearrangement. Your book does a pretty good job of describing how there are fewer gamma delta T cells because they have to rearrange both gamma and delta successfully before a successful alpha rearrangement in order to become a gamma delta. So that's why such a small percentage of T cells become gamma delta, because they have to have 2 successful gene rearrangements prior to a successful beta chain rearrangement in order to go down that path. And so there are those 4 stages of double negative before they become double positive and then single positive before leaving the thymus. Okay, so if you remember, in the B cells, we had a surrogate light chain that paired with the heavy chain. So we reorganized the heavy chain first and tested it. There were a few chances to reorganize the heavy chain, and a successful reorganized heavy chain starts signaling, and that's how the cell knows that it's had a successful reorganization. And it's the same in the T cell. Basically, once you've reorganized either the gamma delta or the beta chains, a signaling complex is formed, and so you get survival signal. So the basis upon which T cells continue down the development path is by initiating successful signaling resulting from reorganizing a T cell receptor. And the result of not getting effective signaling is staying at that stage and continually trying to reorganize a T cell receptor until you run out of options. So again, just adding another level of detail in the last figure we looked at, the beta chain rearrangement occurs during the middle two stages of the double negative in the thymus. If that's happened successfully, you start signaling, and you can pass this checkpoint to do alpha chain rearrangement. If not, you take one more chance to reorganize the beta chain and then die. If you successfully reorganize the beta chain, then you have multiple chances to reorganize the alpha chain before reaching another checkpoint in which you have to have successful alpha and beta to continue. And if you aren't able to do that, then there's not enough signaling and the cell is apoptosis or die. Go ahead.
Why is it that you get to try one more time, not more than one time?
Well, so with the beta chain, it's described molecularly in your book, but it's just the number of genes that are available. So with the beta chain, you have just 2 copies to work with on each chromosome, and the alpha chain, there are more copies to work with. Okay, so I find it easier to remember fewer things rather than more things. So it's easier for me to think about how similar the processes are for T cells and B cells. And this is a complicated diagram, but it just shows that those stages we've been talking about for the T cell have very similar stages for the B cell, and the same requirements occur in for the early B cells. You do DJ recombination followed by BDJ of the heavy chain, and then you go to the light chain. And the other cell surface marker expression is changing throughout, and it's the same for the T cells. The only exception being that there's an early branch point where they decide whether to become gamma delta or alpha beta. And for now, the basic functionality of alpha beta and gamma delta T cells is pretty similar for now. We may eventually talk about there's some differences in what tissues they prefer, but it's basically just expressing TCR from a different set of genes. And you may remember last time that the gamma and delta gene loci are inside the beta chain loci, so they have to reorganize both gamma and delta before beta happens, or the gamma and delta get spliced out, so they'll never have a chance to become gamma and delta. And evolutionarily, why that occurred, I don't know, there must have been some advantage to it. So again, all the events we've been talking about so far occur in the thymus. So the pro-T cell arrives in the thymus. It has receptors for KIT ligand, which is a hematopoietic growth factor, and expresses CD44, and then, which was one of the earliest markers of the T cell, and then enters into the thymus, goes through the gene rearrangement we just talked about, and then in the late phase, there's the positive selection, so testing of the T cell receptor, and that selection has to happen within certain bounds. It has to be successful enough that the T cell receptor signals and keeps the cell alive. But if it's if positive selection has too high an affinity for self, then it's eliminated as a positive selection. And then it exits into the periphery before being able to go through an immune response. So I'm sorry, I'm banging on the same drum over and over again, but it's there's just a lot of different ways to look at the same thing. And just, I don't think we don't even have to go through it again, but basically just showing where the bifurcations are for the gamma delta decision is early and the CD4 CD8 decision is late. And they both come from the same committed progenitor cell. Go ahead, Kai.
In the previous slide,
Uh-huh.
natural killer cell, like how does that come into play?
So natural killer cells actually develop in other tissues, not in the thymus. But there's a common progenitor that's involved. So we're going to have a whole lecture later on in the semester about natural killer cells. Okay. So again, alpha, beta, gamma, delta. So this actually isn't very accurate because it doesn't show the gamma and delta within the beta locus. But in order to be the gamma delta T cell, you have to successfully rearrange both before the beta chain reorganizes, because you don't have to have a successful alpha chain reorganization yet. If you get beta chain reorganization, it complexes with the pre-T cell receptor alpha chain and you get signaling. So there's much more advantage to become an alpha beta T cell. So basically because of the limited number of beta chain constant regions, you only get 2 chances to reorganize the beta chain, whereas alpha chains, there are many more copies available, so it can do multiple rounds of attempted alpha chain. So the beta chain rearrangement happens quickly. The alpha chain, it can wait for alpha chain reorganization for quite some time. Okay, so all of these complicated interactions happen in 3-dimensional space within the thymus. So, and there's quite different cells in different parts of the thymus. In the cortex, there's a lot of cortical epithelial cells and hematopoietic progenitors, whereas in the medulla, there's a lot of antigen presenting cells. So during the process of T cell education, there's a movement from one part to another. So the cells arrive in the endothelium in the medulla, where they are supported in growth factor expression to proliferate, and then as they pass into the double negative stage, they go out into the cortex where the epithelial cells are, so that they can be exposed to all the available antigens for T cell education. And then they pass back into the medulla, into the circulation once positive and negative selection have been effective. So that positive selection, so successful T cell signaling, occurs on those specialized cortical epithelial cells, and I'm not actually sure, we may get to it a little later. Those cells have some very specialized characteristics in that they can express both class 1 and class 2 MHC. So unlike most other cells of the body that aren't antigen presenting cells, they can express class 2. So as many possible peptide MHC combinations as can be expressed are being expressed in the cortex. And if the T cell goes through all those gene rearrangements and doesn't get enough signaling through the T cell receptor, then it'll die. If it gets a little bit of signaling, then it passes on to negative selection. Okay, so because the cells are double positive at at this point during positive selection. They don't know if they're going to be helper T cells or cytotoxic T cells. They can, that TCR that they reorganize can pair with either CD8 to get peptide presentation from class 1 or peptide presentation from class 2. So only one of those has to be successful, and whichever one is more successful determines which type of cell that it's going to become. And in order for all that positive and negative selection to occur in the different parts of the thymus, the T cell has to sequentially interact with different types of cells. So when the thymocyte arrives from the bone marrow and then goes through all those early stages to get to the double positive stage, then it can interact with class 1 and class 2 on the epithelial cells, and then go on to interact with antigen presenting cells during the positive selection phase. So negative selection occurs on the thymic, positive selection occurs on the thymic epithelial cells, and then negative selection on professional antigen presenting cells. And this is just reiterating the same point that professional antigen presenting cells mediate negative selection. Sorry, I'm getting screwed up myself. So in order to successfully rearrange the T cell receptor genes, there has to be some signaling that's positive selection on the epithelium. And then in order to ensure that it's not autoreactive, it interacts with professional antigen presenting cells, in which case tight binding results in death. So in the first stage, death results from not enough signaling. In the second stage, death results from too much signaling. Okay. And so one of the really unique parts about the thymus, those specialized thymic epithelial cells, is that they have some unique molecular tools to express as many self-antigens as possible. So it's mediated by some transcription factors, the AIRE complex. And this is just a demonstration of looking at how many different proteins one can identify by proteomic approaches within the thymus. And you can see that almost every tissue in the body, some proteins from almost every tissue in the body are expressed by the thymic epithelial cells, mediated by this transcription factor. So there's every attempt made to expose those cells to proteins from all our cell tissues in order to have successful positive and negative selection. So, and that expression of that transcription factor is what distinguishes the thymic epithelial cells in the cortex and in the medulla. So, from the internal epithelial cells have less promiscuity of gene expression, whereas the cortical ones have much more. And during the different phases of T cell development, as it moves through different parts of the thymus and gets exposed to more and more cell proteins, the T cells themselves have different thresholds for signaling. So early on in development, you need just a very small amount of signaling to get selection. So you need a very high threshold to get eliminated, whereas later on in survival, extremely tight stimulation is required to eliminate by positive selection. So the specifics of that aren't important, just to point out that there's not one fixed threshold for negative and positive selection, that it changes during the course of T cell development. And I think this is just a repeat of what I was talking about, whereby there's basically a loop of development for the T cells moving out from the medulla into the cortex and then back into the medulla, and with negative selection followed by positive selection. And this is a point that we talked about earlier in the semester, just that the thymus basically decreases in cellularity during the course of one's life, and our ability to mediate T cell mediated immune responses declines with the kinetics of that loss of thymus. Okay, so any questions on that?
If you, I guess, were exposed to like lots and lots of different pathogens, I guess, or like over the course of your life, would your immune system maybe stay up and function a little bit more as those cells stay there, or I guess you could just.
I'm not aware that the rate of T cell development and action of the thymus depends on how much infection you're getting. What's going to be affected is the kind of T cell responses you have and the kind of memory you get. And we started talking about that in your discussion last week. And the one part that I wanted to get to was the hygiene hypothesis says that we get allergic responses because we're too clean, we haven't had enough infections. But that's not the whole story. It's what kind of infections we have. We have to have infections of a kind that cause Th1 mediated immune responses, which we're going to get to in the next lecture, rather than Th2 immune responses. So, because both result in memory, it's the Th2 memory that leads to allergic responses. So, I think that how much infection one experiences, unless it's to a degree of affecting your total health, doesn't change T cell education. It changes memory. Okay.
Before, it's like a couple lectures ago, you mentioned that like at around puberty, your thymus stops educating T cells. That's right?
It's not quite so
Why is it?
finite
Okay.
as that. It's a continuum, basically like this. So
Does
sorry, go ahead.
Does that also mean that like T cell production stops?
No, so you keep making T cells. And there seems to be some kind of peripheral education that goes on, but nobody really knows exactly how that occurs. I mean, definitely, if you take a thymus out of a newborn person or out of a newborn animal, then there's actually DeGeorge syndrome where people are born without a thymus and they are horribly immunocompromised. But somehow the gradual loss of thymus later in life doesn't result in an end T cell education. But what's hard to tell at that point is how much of your T-mediated immunity is coming from existing memory, from early educated T cells.
Which
How
are
much
just cloning themselves.
Exactly.
Basically. Okay.
So, and they can do that experiment in mice, take out the thymus at different ages, get rid of all their T cells and start again and see how well. And certainly you can get T cell education peripherally in adult animals, but nobody really knows exactly how that happens. But it's assumed that most of your important T cell mediated immunity in later adulthood is coming from earlier educated
Just making
T cells
clones
that
of
are
themselves.
memory, yeah.
And does that cloning have to happen in the bone marrow, or can it happen anywhere?
So most of it happens in lymph nodes, actually.
And
So,
so that-
yeah, because that's the site of T-mediated immune responses. And that's what we'll talk about next lecture. So, at the end of the B-cell development lecture, we talked about how not the positive and negative selection isn't perfect. You do get autoreactive B cells. And so there are mechanisms to maintain tolerance of autoreactive cells. And it's just, and the same is also true for T cells. T cells that that are exposed to self-antigen are maintained in an inactive state by suppressor T cells. And so we'll have a whole lecture about immune tolerance, but there's a specialized type of T regulatory cells that actively suppress the activity of T effector cells that are autoreactive. And that occurs when they're both being presented antigen by the same antigen presenting cell. Go ahead, Pat.
Is there any chance that a more like innate immune system can have like autoimmunity disease? Or is it like unlikely? Because
So, I mean, can a cell of the innate immune system mediate autoimmunity? So, innate immune cells participate in autoimmune reactions because when a T cell mediates direct cell killing or mediates autoantibody production, the effector cells, phagocytes, etc., are responding to those immune complexes and mediating. So certainly the innate immune cells are pretty good in general at identifying self and not reacting to it. But they are highly activated by T cells and autoreactive antibodies. But like many things in immunology, it's not perfect because there are, when you look at big genome-wide association studies, there are genes involved in innate immunity that do seem to predispose to some autoimmune diseases. So an example is there are mutations in TLR7, which is an innate immune receptor for RNA and DNA that seems to predispose to lupus. So in lupus, the autoimmunity is against nuclear contents, nucleic acids. So the thought is that that mutation changes the threshold for activating TLR7 with endogenous nucleic acids instead of foreign nucleic acids. So it's not, so in general, autoimmunity is an adaptive immune response, but to say that the innate immune system doesn't play a role would be untrue. But the innate immune system in and of itself cannot mediate autoimmunity without the specificity of the adaptive immune response. So there are, for example, there are all kinds of gene knockout mice, so mice that don't have T cells, mice that don't have B cells, mice that don't have T cell receptors, et cetera, and none of those mice can be induced to have an autoimmune disease in the absence of an adaptive immune system. Okay. So this is just another look at the same picture we've been looking at all lecture, basically showing that negative and positive selection happen by T cells interacting with different specialized cell types within the thymus before a T cell can be successfully developed and pass into the circulation. And this occurs for less than 10% of the T cells that enter the thymus as progenitors. So it's a highly inefficient process, which is why the T cells take as many chances as they can to successfully reorganize their genes before starting with another fresh cell. And obviously, it's critically important because of the potential for autoimmune responses. Because the specificity of the T cell is just to a short peptide combined with MHC, education is critically important. That's it. I'm happy to answer any more questions about T cell development. Let me take 5 minutes for the presenters to get organized for the discussion. And we'll have the discussion and have a longer break afterwards. And then the second lecture is on the actual T-cell responses after development has occurred.
We had a lot to cover, but I didn't have to narrow it down because it's such a wide topic.
Hi.
Head out after this session so I can not drive home all the way in the dark.
Sorry?
It's okay, I have to head out after this
Oh,
session.
yeah. No problem.
Something I ran into last night when I was working on the take-home that ended me being less specific than I wanted to be. Because one of the chapters here, this says that dendritic cells carry all the TLRs except TLR9. And that's like see chapter 2. But when I looked at the table that I assumed they were referring to, they only carry a handful of TLRs. So I wasn't sure if I was missing something. Because right here, it has like all the TLRs listed in like cells that are carrying them. And, you know, it's clear that there's a bunch that dendritic cells don't carry.
So the problem is that it's not telling you that there are different kinds of dendritic cells.
Yeah, it has the one type of post, whatever, post. Plasmacytoid.
Right, so there's myeloid dendritic cells and lots of subtypes of those. Most have a handful of them, so that all the dendritic cells cover all of them, but no one individual
Carries.
will have all of them.
That's what I figured it meant, but then it only listed the one subtype. That's the one I really saw mentioned in the reference. Okay, thanks. To be so there was I wasn't sure. I wasn't sure if someone was playing. That's fine. It doesn't matter. So I'm just saying. Yeah, somewhere. Okay. We're fine. Yeah. So I'm just running through
It's sad.
that's how I do it. Yeah. So like no donations. So I was into, did you guys look at the basic people?
So great.
There was like only one that was like 2011 Berlin was stem cell research board. I was actually with it in July of last year in Boston for women. And so far they've shown a sign of HIV that they moved away from before they were able to transfer something else last year. It was all stem cell research, and it was basically they were saying that the stem cells were able to go into the bodies they were being donated to, and they were actually they think it is like differentiating and proliferating enough and actually getting rid of the entire lymphocyte population of the entire infected lymphocyte population, so they're just completely replacing it. And so with blood donations, like you coordinate or for whatever reason. They could do anything
Make sure it's like I don't know, this was literally kind of like they were full and then one was just something else. And all of it's kind of hard though, because we forced to do Elliot.
Wait, can you guys all
Yeah. And I was like, that's really not safe. Like, I have two PCs, like, they're just like, yeah.
but like basically
Oh, they lined up today. And she knows who did it. We got it. We did all of our freshmen applied.
Yeah, I was like five.
I did that too though. I go to myself.
I mean, I don't think all the parents and stuff are just different, but a majority. I don't remember yeah, like, it's swaps, they're like, talking about you, like, dude, like, that's the first year life. That's awesome. Like
She has a sketchbook.
I think they'd appreciate it. Yeah, good thing I can't be.
Look, I highlighted them, I like read it.
Oh, wow.
My
That's what they're
We have to do like at least
No, I mean, like, I don't care, kind of. No, I did not have unsafe.
Help a girl out.
By the way, no, I had this like sunburn, and then I had like this like
Sounds about how it's like
spots on my body. So I was like, why? Like, it was like
But you can always try to
curious how this.
things you learn.
No, you're just being phobia. I'm like, no, I still
just
feel
load the
it.
YouTube video on another tab as well. That's probably, I found one that's a minute long, that goes through
Yeah,
quickly.
I was just a little crazy.
The link is in the,
Oh.
it's on the slide. If you go to, the bottom of that one, no, it's all the way in line one. Top ones.
I don't know what that's yours, right?
Top the first ones. That was me.
No one thought it could happen in North America.
Yeah. One of those. Yeah. The other one's, this one's way more intense. This one's 5 minutes long and it has one of those like soothing commentators on it and everything. But that was way too long for this.
Which is probably like 5 minutes.
Okay, we're gonna start, if everyone's okay with that. For those of you who didn't even look at the article, we're doing our presentation on HIV/AIDS and the immune system. So just a quick overview of how we're structuring this. I'm gonna start and go through how HIV gets inside and then infects the body, and what the implications are for the immune system through that. James is gonna go through what the stages and symptoms of HIV are. Emily's going to talk about what the current picture of vaccination and treatment looks like. Tra is going to look at how that affects the presence of HIV and AIDS today. And then Becca's going to talk more about the specific genetics of HIV and how those genetics are going to be possible windows into cures and vaccinations for the future. So just as far as how HIV enters the body, a lot of you guys probably know this. It enters through any fluid besides saliva, so sexual fluid or blood, unprotected sex, dirty needles, anything like that, tattooing as well, anything that really can actually get inside of your bloodstream. So as one of the things I was reading last night says, if someone spits on you that has HIV, unless they're bleeding, you're not going to transmit anything. There's no evidence to show that you'll actually get something from saliva. And it can't enter through any simple contact like that. We have a short video, you just do that now. It's only a minute long, and there's a longer one that has the link at the end. So this is just a really quick overview, but there's a 5-minute one that's more informative that's below it as well.
As a result, a heterogeneous viral population containing viral variants able to withstand immune and drug pressure evolved to
That's terrible. We can click on the other one quick then, because it gives a quick, you can just skip through it. So I'm on the wrong link, ended up in there, because I looked at a bunch of them.
Targeting HIV replication. One is the infection of a suitable host cell, such as a
Just
CD4
leave it here for
positive
now,
T
and then I'll
lymphocyte.
talk through it in a second. But
Entry of HIV into the cell requires
This
the
goes really
presence of
slowly,
certain
though.
receptors on the cell surface.
So Becca's going to talk more about the receptors
CD4
here as well.
receptors and co-receptors, such as CCR5 or CXCR4. These receptors interact with protein complexes, which are embedded in the viral envelope. These complexes are composed of two glycoprotein earned attachment.
That's one necessary. I'll talk to you that in a second as well. But basically, it's able to fuse the membrane of the lymphocytes there, and then the membrane's able to fuse the two things so they can share all their intracellular
strands and 3 essential replication enzymes.
How did they film that?
I don't know.
Protease and reverse transcriptase.
So those are the 3 kind of important things that I'll go through, as well as just the 3 proteins that HIV enters the body with that allows it to work through. So it attacks T cells, primarily CD4 T cells, and it binds to the CD4 receptors as well as a co-receptor, as you were seeing at the beginning of that animation. That allows the 2 membranes to fuse. Once the virus is inside, the reverse transcriptase protein that the HIV virus carries basically makes half of a DNA copy off of the RNA single-strand RNA that it carries as DNA, and then that is then copied. So the integrase installs the new DNA. Basically, there's the RNA single strand is paired by the reverse transcriptase, then the RNA strand is broken down, and a new DNA strand is paired up with the copied one off of the single strand RNA. The integrase is able to install that, goes into the nucleus of the infected lymphocyte, the T cell, installs that into the DNA of the host cell where it can lay dormant. Once that lymphocyte is activated, then it's replicated along with the rest of the DNA of the lymphocyte. So this allows all the viral proteins also to be replicated. These are broken down by a protease that the HIV also carries into smaller bits that can then be lined up on the membrane of the lymphocyte, and then those are joined by another strand of the single RNA that's copied for the HIV, and then those can then bud off from the lymphocyte, forming new immature viral cells, and then those, once they mature, are able to go off and infect more lymphocytes. So as James is going to talk about, you can see once it's inside, integrated into the DNA of the lymphocyte, it doesn't necessarily have to be activated right away, not until that specific lymphocyte becomes activated. And then I'll talk about how that kind of affects us, what the implications are for there. So just quick review again. The CD4 T cells produce cytokines. It's one of their main functions, and they assist macrophages with T helper cell 1 type and B cells as well with helper 2 type. And those join the immune response, and a couple of those kind of cascade leads to inflammation and antibody production, so opsonizing antibodies from macrophages and neutralizing antibodies from B cells. There's other things that those lead to as well, the cytokine responses, but that's kind of a general overview. So basically it infects the helper T cells, it uses them to replicate itself, and then it ends up killing them as well afterwards. So you end up infecting a bunch of T cells, and then as those T cells are reacting, you're producing more of the virus, and you're also losing T cells because as the virus infects more, it's eventually killing them after it uses them to replicate. So that means you have decreased T cells in general, CD4 type, which means a loss in immune response overall, and you also have a lack of signals via the cytokines of the CD4 cells to activate macrophages and B cells. So you just lose immune response in general over time. So again, the longer it's kind of active in your system, the more you lose.
Okay, so these slides are on the symptoms of HIV and AIDS, and having HIV and AIDS can lead to various illnesses, cancers, and neurological problems. But HIV infection actually takes place during 3 stages. The first stage comes right after the initial infection, and it occurs 2 to 4 weeks after that infection, and a lot of people suffer ARS, which is acute retroviral syndrome, which is the body's natural reaction to the virus, and it presents itself with flu-like symptoms like fever, swollen glands, sore throat, rash, fatigue, etc., etc. And then the second stage is actually a period of no symptoms, and this is where a lot of people actually who don't know that they got infected with HIV transmit the disease to other people. And it's where it's called the asymptomatic period, where the immune system has succumbed to HIV as the HIV slowly kills the CD4 T cells. And then the third stage occurs when the CD4 T cell number drops below 200, and this is the point where doctors can diagnose the infected person with AIDS.
Can you tell if somebody's HIV infected before their T cell count drops before 200?
Well, yes, because, well, I don't know because even if it's above 200, I don't know if CD4 count only specifically applies to HIV infections. So if it's still above 200, I'm actually not sure.
What do you guys think?
Well, are you asking physically or are you asking?
Is there a blood?
Diagnostically.
Diagnostic.
You, I think
Okay.
you
There's a
can,
blood test for it.
yeah, you can test
Yeah,
and you can
there's-
do a
there's
cheek
definitely-
swab as well.
Right.
For presence of the HIV
Right,
virus.
so anyone who may have had an HIV test, they're looking for antibodies against HIV. And the more sensitive test they have now is actually PCR when they're looking for the actual genes of HIV. So you can definitely tell if someone was infected long before they have AIDS. And that's the ideal situation, right? If you can start treating before loss of T cells.
Right.
And what do first responders do if they are exposed to somebody whose HIV status they don't know?
You have to wait for a short incubation period, and then you have to go get tested.
Yeah, but usually if you can't get the HIV status of the person whose blood you're exposed to, you start taking antiretrovirals before you even know whether you're positive. So, because that's actually shown to prevent an establishment of infection. So during this first stage, you actually prevent the infection. Merritt?
How soon after being infected can a person be diagnosed?
Well, it has to be after the first stage, once you've made antibodies, you get that expansion. But I don't want to ask these guys the question.
Can I ask it?
Yeah,
Oh,
ask
so
them.
is it possible that, do any people, you're talking about antiretrovirals, is it possible, can the immune system destroy HIV? Like, are there rare cases when people have a strong enough immune system to get rid of it?
Well, there's genetic resistance, which is just in some populations of people, depending on just evolutionary exposure to some degree. There's been one case of a cure in 2011 in Berlin, and there's 2 more in Boston currently. And those are all through stem cell treatment. So there hasn't been any cases that I'm aware of where someone's immune response has been strong enough to fight it off, but there are people who are genetically resistant who could theoretically have been exposed to it and fought it off, but since they never were diagnosed or never had to find out if they were HIV positive or what have you, then we wouldn't know that for sure.
And we're going to go into the genetic resistance in more depth later on. Do you have a question?
Well, I kind of have something to add on to that. I don't know if you guys look into the retroviral load, because they use that also as an indicator of HIV. And aren't there instances where you take your antivirals and then your retroviral load is so low that it's considered to be undetectable, and so they consider you to be cured, even though they know you still are infected with HIV?
Mm-hmm.
So isn't that an in-between where it's a cure, but it's not really a cure?
Yeah,
Because-
actually, in some of the cases that we were looking at, there's, I think the Boston patients, there's 2 or 3 of them they consider fully cured, but there's also people who are no longer showing significant viral load, and they have to wait, I think the study said a year or so, and continue testing viral load to be able to tell if the virus is just reacting to the treatment or if it's actually been eradicated from their system.
Yeah, isn't there a fine window where it's if you take your antivirus 80% of the time versus taking your antivirus 90% of the time, and that window between 80% and 90% is where you develop immunity, to your antivirals, where the medicine stops working for you? I don't know if you guys looked into that, or if you guys found any studies about that.
I didn't know anything about that, but
That's how. Okay, I was just wondering if you guys know of any other cases about developing resistance towards the drugs, towards the antivirals.
Yeah, I talk about antivirals a little, but not so much about the
resistance towards
yeah,
it?
resistance
Okay.
to it.
I know in one of the papers that I read, it talked about patients needing to like switch their drug cocktails occasionally, which is probably what you're talking about because they start having reduced efficiency, or they like their viral load starts increasing when it should be decreasing. So I guess that's probably related.
Good questions.
Yeah. So in the late stages of HIV and the transition to AIDS, people are going to experience symptoms of rapid weight loss, recurring fever, extreme tiredness, night sweats, long durations of diarrhea, and purple spots on skin, among other things. And opportunistic infections occur as a result of a weakened immune system. Examples include pneumocystis, pneumonia, and tuberculosis, but different conditions can occur at different stages of the HIV infection. And they're caused by a variety of germs, bacteria, viruses, protozoa, or fungi. In the first stage of HIV infection, a person can develop tuberculosis, malaria, bacterial pneumonia, herpes zoster, staph infection, and septicemia. And then it gets a little more serious in the third stage of HIV infection, and one can develop fatal infections such as PCP, toxoplasmosis, and cryptococcus. And maintaining a stable CD4 count will decrease the likelihood of contracting an HIV opportunistic infection.
Do you know why the infections of the first and the third stage might be different?
I would guess because in the first stage, well, it's the HIV. It's pretty new to the body, so your immune system is still somewhat intact, and so you still have an immune system that can fight off these infections. But in the third stage, your body's has already succumbed to the HIV virus after the asymptomatic period. So,
Yeah.
Also,
Go ahead.
yeah, I'd say that assuming that if your CD4 T cells drop way below and you're an adult and you have HIV, then you're also done educating T cells. So if you start losing more and more T cells, you're going to have greater probability of losing whole groups of T cells. And so as you lose more groups, you're susceptible to more diseases.
Yeah, so that's true what you guys said. I'm not sure that these three stages of the infection are the same as on your next slide, the stages of, are those the same stages in terms of?
yeah.
Okay, because, well, what I was actually thinking of was the fact that many of us are chronically infected with herpes zoster, which is what causes cold sores. We all usually have staph and streptococcus on us. So basically, those organisms are on us, and just a little tip in the balance of the immune system, they can take off. Whereas the later ones, toxoplasmosis and cryptococcus, those are in the environment. We aren't chronically colonized with those, so you have to actually pick it up from your environment. So,
Okay.
did you have a question, Jenny?
Oh, no.
Okay.
Stretching.
Those can be said for tuberculosis too. A lot of people are like walking around with dormant tuberculosis that just are like your immune system is strong enough to fight it or keep it at bay, but as soon as it's like, infected in some other way, that can come in too.
Right.
Right, does that like indicate that like even in the first stage the immune system's already like been taken down by the infection? If like a small dosage of like any of those bacteria or pathogens can cause those.
Well, you could think about all of us who are healthy with full immune systems get pneumonia, have cold sores. So just a little tip is enough to set those infections off. So I think it would indicate that if those things are happening at that early on before you're losing CD4 cells, that HIV is having an effect on the immune system.
Is it possible to simultaneously treat someone for HIV as well as an infection caused by having it?
I would imagine
Yeah, I would imagine so, yeah.
Especially if it's a bacterial infection, you could give them antibiotics. It's just, I think it's really just a matter of the fact that the immune system is being suppressed. That's why they're suddenly acquiring these infections.
And a drug that you can actually treat opportunistic infections is cotrimoxazole, which is a cheap and widely available drug that prevents a lot of different kinds of opportunistic infections. And it's actually been shown to reduce mortality rates among HIV-positive people initiating retroviral therapy. But why is prevention of opportunistic infection important? For the longevity of HIV-positive people, obviously, and to prevent the spread of transmissible opportunistic infections, especially because these infections occur at a high rate among HIV-positive people. And how we prevent them is to reduce exposure to germs. So staying away from uncooked meat, domestic animals, human waste, water.
Like water.
Wild water, yeah. Right. Unpurified water.
So there's currently no vaccination for HIV. However, there are a few reasons for this. A normal vaccine for any type of disease increases the number of B and T cells directed toward a specific virus. However, HIV is a retrovirus, which means it can use its RNA to make DNA. And the enzyme that does this is reverse transcriptase, which is not perfect, and it doesn't have a proofreading ability. So a lot of errors are made while it's being translating RNA to DNA, and so there, as a result, a lot of strains come out of it. And it's hard to make a specific vaccine because then you have to decide you can only make it towards one strain, and since it's like changing constantly and continuously, it's really hard to do that. It's very rare for two people to have the same type of strain of HIV, so it's kind of hard to decide where to, like, in what direction to research everything. Also, most other vaccines are against the disease, not the virus, and HIV is the virus causing the disease AIDS, so it's kind of hard to make a vaccine specifically for the virus. With that being said, there are some clinical trials that have been done to try to find a vaccine. Most have been unsuccessful. In 2009, I think, the most successful one was the Thai trial. It's RV144 trial is also what it's called. They used a combination of two vaccinations that had failed in previous trials. They found these vaccinations these strains of HIV virus were very common in Thailand, so they decided to combine them and see if that would work. So they injected a group of people with the vaccine and then let them live their lives. And every year they would be tested to see if they had it. And for those who came in contact with the virus during that time, there was 30% lower than the rate of HIV infection in volunteers who received the placebo. So it was significant, the amount of people who had the vaccination and didn't get the virus even after being exposed to it. So they're doing more research into that, but nothing is concrete yet. And no vaccine is really, it's unsure if that would actually work in the United States or anywhere else because it's specific to Thailand and the strains that come there.
I'm sorry, I think I'm confused a little bit. They injected people with HIV after giving them the
No.
vaccination?
They gave them the vaccination, and then they let them live, and then some people were exposed to the virus.
No, so how did they know if they were exposed? I guess that's what I'm confused about. How did they know? So did they go off and intentionally expose themselves to the virus?
It was a group of I think it was 20,000 people that got the vaccination in the first place. And then I'm not sure how they could tell that it must have been that they were tested for HIV, and then I'm not sure how they could tell. There was a total of like 200 of the people were exposed to the virus during that time. I'm not sure how they knew that it was like if they didn't actually get the virus, how they knew that they were.
It's just straight numbers. That's why the
Yeah.
trial has to be so big. So you enroll 20,000 people. Historically, they knew 200 of whatever number of that number of people in Thailand would, through letting them live their lives,
Yeah.
become HIV infected. And half of the people had been vaccinated and the other half had just gotten an injection with vehicle. So if you had gotten the vaccine, then there was 50 instead of 100, and if you hadn't gotten the vaccine. So basically, you have to do a gigantic study in the wild and compare the rate. You don't actually know how many people got exposed, you just know who ended up with HIV in the end, and say the exposures were roughly equal between your two groups.
So do they just like tell them to come back like 10 years later, come back and then get tested again to see if they were
Yeah, I
exposed?
think they were tested like every 5 years or so.
I know in other tests or in other trials that have been done, they also target high-risk groups, so they'll recruit people who are drug users or are like sex workers or something, which increases the chance that they'll be exposed to HIV in their lifetime.
Yeah. So, but also there's a drug called Truvada, which has shown to have some preventative measures, which might be what you were talking about with if someone's exposed to someone who questionably has HIV. But this is like a combination of 3 antiretroviral drugs, and it's often used as a treatment, but there is some preventative measures especially.
In the trials, you mentioned that they tested them every 5 years.
Mhm.
Was there a decrease in, I guess, like did the vaccine wear off? Do you know? Like, or did it have sustained?
Not that I know of, but I'm not entirely sure. I didn't find anything that said that.
Can you have the last slide, please? So can strains like within the actual body evolve? Or I mean, so is that what makes it so hard for the immune system to fight it in the first place? Because it can't really create like an antibody for that is effective?
Yeah, as it's replicating, it's changing constantly, and they usually, as she was saying, they don't find a single strain present in 2 people, and they usually also don't find just one strain present in people who are infected. It's whenever there's the RNA being replicated, when the virus enters lymphocyte, it can have errors and mutations and then transform, but still maintain its properties of entering lymphocytes and doing everything else it does.
And I think the first line of your slide is key too, because we haven't had our vaccines lecture yet, but the best vaccines induce robust T and B cell mediated immunity. And what cell mediates T cell and B cell mediated immunity? CD4 T cells.
Yeah.
So it's exactly the cell you need to work the best to have an effective immune response is being impaired in this, plus it's a retrovirus, plus it's evolving constantly.
So right now the treatment for it is to take antiretroviral drugs, and there are 5 classes of these drugs. They all have to do with how HIV enters the cell and inhibiting some part of that stage. The first is entry fusion inhibitors. So when the virus is entering the stage, it will either block the receptor on the virus or on the CD4 receptor, and not allow it to enter or fuse with the cell. The next is nucleotide reverse transcriptase inhibitors, also called NUCs, and these block the reverse transcriptase. They attach to the DNA that's or the RNA they're trying to build DNA off of, and won't let the reverse transcriptase continue to replicate. And then there's non-nucleoside reverse transcriptase inhibitors, and these connect to the reverse transcriptase and won't let it attach to the RNA at all. Then there's also integrase inhibitors. So integrase is the enzyme that puts the newly made viral DNA into the host cell DNA. And the integrase inhibitors will connect to the enzyme so that it can't enter. Then protease inhibitors. Protease is the enzyme that once the virus's DNA is copied and is leaving the cell, will cut up the virus so that it can spread out, and it'll inhibit that enzyme. Most people take a combination of at least 3 of these drugs, and this is called highly active antiretroviral therapy. Most people take 2 classes from 2 of the reverse transcriptase ones, either NRTI or NNRTI, and 1 from protease inhibitors. That's the most common. I'm not sure why they decided. I couldn't really find what that was why those were the best options, but I think it has to do with developing resistance, and of course you want 2 different classes so you don't develop resistance against everything, or if you only take 1 drug, that will probably develop resistance pretty quickly, but I'm not sure why those 2 are the most commonly used.
Okay, so as you know, the medical management of HIV is evolving. Currently, there are more than 25 antiretroviral drugs available for the management of HIV. The success of these drugs means that people who are infected with HIV are now living longer, and it means that they must deal with a series of age-related diseases. So what it is now is that people are now spending more time managing the drug, and more time sort of managing the complications that come along with as people get older, dealing with the disease that are sort of enhanced or influenced by the fact that they have HIV. Excuse me. So it has been shown that HIV patients treated with antiretroviral drugs have increased risk of non-AIDS complication. And this is also associated with the lower CD4 T cells, and the increased risks are in cardiovascular disease, malignancy, kidney failure, liver disease, bone disease, and neurological complications. So, which non-AIDS related disease are affected? So in here I will focus on the 4 main ones, and that is cardiovascular disease, cancer, liver, and renal disease. So for cardiovascular disease, it has been found that the antiviral treatment is associated with early heart disease, even after controlling the age or the traditional cardiovascular risk factors. And the risk for these diseases are much higher in untreated patients than those that are treated because of inflammation, or seem higher in untreated infections. And it has been shown that some antiretroviral drugs have direct effects on cardiovascular disease. For example, prolonged exposure to protease inhibitors is associated with insulin resistance and a higher rate of cardiovascular disease events. Another thing was abacavir is a common used nucleoside analog, which seems to increase the risk of heart disease directly, and it's believed that it's due to the inflammatory effect also. Yeah. Oh, sorry. And yeah, but despite all of that, the benefits outweighs the potential negative effects. Because people do need these drugs to live longer lives. And for cancer, untreated patients have a higher risk of cancer, and this is including lung cancer, skin cancer, colorectal cancer, and prostate cancer. So
For liver and renal disease, liver disease is common in HIV-infected adults due to the high rates of chronic viral hepatitis and long-term exposure to toxic antiretroviral drugs. So in a SMART study that they did that was conducted using 5,472 patients, they found that people with interrupted treatments were more likely to develop to develop liver failure than those who did not. And but however, for kidney failure, there are no difference in terms of risk for those who are treated continuously and those who are treated discontinuously. Overall, the increased risk for age-associated disease is the result of the immunodeficiency and inflammation. So overall, the effectiveness of these drugs also introduces new problems. For example, the care of patient with HIV will become more complex as the patients, they grow older and they confront more unique challenges. So they would need cardiologists, oncologists, and other medical specialists to that would be more on demand, be on call to treat these complex diseases.
Great. So the last topic is genetic resistance. Earlier we talked about how HIV uses CD4 the CD4 receptor to get into the T cell. It also uses co-receptors. One of them is the CCR5 co-receptor. And there's actually a genetic mutation that's been found in certain populations that you can either be heterozygous or homozygous for it. So it creates a CCR5 protein that's significantly smaller and doesn't really extend to the cell surface, so HIV can't bind onto it along with the CD4 receptor, and it makes it significantly harder for it to get into the cell, or it can offer almost complete immunity to infection. So if you're homozygous, that means that you have both the alleles for, I think it's recessive, so you have to be homozygous recessive. You have both the alleles that code for this mutation, and that usually lends the person virtual immunity to HIV infection. Whereas if you're heterozygous, you are resistant to infection because not all of your CD4 T cells express fully functioning CCR5 receptors, but you can still become infected. And this mutation is actually found in descendants of Northwestern Europeans, which is kind of interesting. They think it might be traced back to a single mutation that was spread around the continent by Viking invasions, fun fact. And it's actually absent in most African and Asian populations. It is, however, found in South America and a lot of Native communities as well, which and Latin American communities, which is interesting because it kind of speaks to colonization and mingling of Western. Yeah. Good question.
Do CCR5s have any other functions associated, or has that not been?
I'm actually not sure. I know that they act as a co-receptor for CD4 receptors, but I'm not entirely sure if they have any other purpose.
Do you guys know? So what is it a receptor for, CCR5?
I think HIV attaches to it alongside.
No, but in the absence of HIV, what is CCR5 a receptor for?
I'm actually not sure.
It's a chemokine receptor.
Okay.
So probably nobody remembers the first or second lecture we talked about chemokines, and they're very promiscuous. So multiple chemokines can bind to multiple chemokine receptors, and so there appears to be no obvious immunodeficiency of CCR5 delta people. That they have enough other chemokines and other chemokine receptors that there's no immunodeficiency. But it's interesting, actually, they didn't know that HIV used co-receptors until they discovered these people. They basically had cohorts of people that were doing the worst things you possibly could in terms of IV drug use, sexuality, etc., and never, like, they just had these frequent flyers of people who had all kinds of exposures and were never getting sick. So they actually genetically investigated those people to discover this mutation, and then that caused them to figure, so why is this receptor? This has nothing to do with HIV process that we know about. So that's actually how they figured out the process of HIV using the chemokine co-receptors. And CCR5 is actually not the only co-receptor that HIV can use.
So, because it's most common, I guess I said, it's practically absent in African and Asian populations. So some researchers have actually cited that as a part of the explanation as to why there's such a bad AIDS epidemic in a lot of parts of Africa. And there's actually some studies that have shown differential expression of the CCR5 mutation between men and women. So they have different levels of expression, for example, in the vagina and the anus in women. So it makes them, I think in a study in Yucatan, Mexico, they found that women were actually less likely to contract the disease than men, at least with the CCR5 mutation, which is kind of interesting. So they're still doing research into the sex-based difference in that. So how does this affect people who have HIV or who may be looking for treatment for it? A lot of people have attempted to isolate the genes responsible for this and try to, study them and incorporate the CCR5 mutation into some sort of therapy. And there was actually, like Ryan was talking about, the Boston patients. It's a group of 11 patients where they were born with one non-functional copy of the CCR5 genes, so they are heterozygous. And they're infected with HIV, not injected with it. They got it of their own accord. So they...
The fun way.
Fun way, yeah. So they would remove blood cells from these patients and then genetically engineer them so as to try to eliminate the second functional copy of the gene. So pretty much you create genes that don't have functional CCR5 co-receptor. So they then reinfuse the blood into the patients. And so 3 out of the 7 patients achieved undetectable viral load. Sorry, it's 7 because 4 of the patients had complications that made it so that their results were pretty much nullified. 3 out of the 7 patients achieved an undetectable viral load, which it's a continuing study, so they're going to track their viral load and see if it actually cured them or if it's just, a sudden latency from the treatment.
Do they have continuing antiviral treatment as well, or?
I believe they do, in addition to that.
Any questions in general first, I guess? Does anyone have any questions first? We have some discussion questions thrown out there, but if anyone has anything they want to ask.
Are there any RNA therapies that can works?
Not that I read into at all, or not that I saw, and I didn't see any evidence of.
If you're going to do that gene therapy thing, wouldn't you have to get every type of T cell that you want, or every type of T cell that you want to save and do that process? Otherwise, you could lose whole batches, as we were talking about earlier, right? So you have to get every type of T cell. Is that not the case?
For genetic therapy, where they'd be targeting the RNA of the HIV?
No, sorry, the last slide with the Boston study. Would you need to have every type of T cell that you want to save in that process? Do you understand what I'm saying? Because otherwise you could lose like a whole population of T cells.
I think they just genetically re-engineered the CD4 T cells and then put them back into the patient's bodies.
I think I know. I'm not really sure, Caleb. Yeah.
Ian, did you have a question?
Yeah. So could this be the gene therapy? Could it be kind of like an, I guess, I don't know if atypical vaccination would be the right word, but could they, or could you like kind of introduce the deficient one like early in development to people before they have HIV, so that they're resistant, kind of like the people with the natural mutation have it?
I'm actually not sure about that. I know it would have to depend a lot on whether, like, you'd have to find a perfect match for that person with blood type and who also has the mutation, so it'd be pretty difficult to actually achieve, but theoretically it might work.
It's kind of what I was getting at before the discussion question too. It's
Yeah.
like possible blood therapy in the future based on these studies, but I don't know.
I don't think anyone's done any like conclusive research on whether or not that would be
Yeah.
effective.
The Boston study was last July, like 2013, and they were like, we're gonna follow them for a year and see what happens. So it's very new. So there's still a lot of ideas being developed. There's not a lot of research or material out there right now on that sort of thing.
Do you have a question?
Yeah, do you guys know what Truvada is and why, like, I've heard of people being, like, pre-exposure to antiretrovirals actually can help you once you're exposed to HIV, but I don't know if that's the same thing as Truvada or not.
Well, it's a combination of 3 antiretroviral drugs. But well,
I think that's how it's
yeah,
using
that's
it. I
what
just didn't know the
it's
name of
saying.
it.
Yeah. Yeah.
For the gene therapy, do you have to catch those patients at a certain point in their infection? Like once you get to a certain point, is it irrecoverable, like the amount of T cells that you have? Because, I mean, even after like a certain point, you change the ability of the virus to attach, or haven't you already, like I said, at a certain point depleted your number so badly that it doesn't matter?
Yeah, I think you have to, for theoretical gene treatment, you'd have to probably attack the disease while it's in like the intermediate stage, because you're right, by the time that you develop AIDS, your immune system, like your CD4 cells have already been taken over mostly or killed. And so that the treatment would really focus on reducing the spread of HIV throughout the body. So at that point, you're right, it wouldn't do a whole lot of good, I don't think, if you've already developed AIDS. Any other questions? Cool. So we have some discussion questions. If you guys want to take a crack at them. Or I don't know how we're doing on
This is
time.
good discussion. We can take 5 or 10 minutes and then we'll take a 20-minute break or something.
Okay.
So number one, we kind of already answered a little bit as far as how it mutates. So I guess, do you guys have any thoughts into, this is obviously an impossible question because it's what people are studying, but a lot of current treatments and vaccinations we were talking about is all the antiretroviral stuff. So about blocking HIV from getting in, but obviously once you're getting treated with these, it means HIV is already infected a good portion of your CD4 cells, so there has to be a better way to either eliminate those CD4 cells besides just the HIV killing them while also preventing the HIV from binding to new ones. So do you guys have any thoughts on how there might be, or new ways that they could try and prevent spread or anything?
Asking a room of college students to solve
Yeah.
the AIDS epidemic.
Well, just ideas,
No
yeah, I
pressure.
Because obviously it's a really complicated issue, especially because of the mutations. That's what makes it extremely difficult. And as we were talking about, it targets kind of the one cell that really boosts your immune response. So.
Ian, did you have something?
Yeah, I think that probably the answer would lie in, well, what I was saying before, now that I think about it, like, isn't practical, but like, maybe like altering gene, I mean, this is kind of way in the future, but like altering gene expression so that the transcript, or altering somewhere between making the protein for this, was it CCR5?
Mm-hmm.
Somewhere between like blocking the ultimately like making that would probably, like altering some level of gene to protein would probably be the best idea, because that way you don't have like the toxic side effects of it, like antiretrovirals and whatnot.
I was just wondering, like, do you think, like, evolutionarily, like, we could eventually not rely so much on the CD4 T cell and, like, you know how, I don't know, I feel like there's a lot of like, our immune system's so complicated and there's so much like backfall on certain things. Like if something happens to something, like, I don't know, for like the example we're talking about the MIDAA, like something happened to that, like there's always like things that you can fall back on because like the immune system has like other methods of helping ourselves. So do you think like eventually we
Well,
will?
so prior to antiretroviral therapy, no one survived AIDS. So that's obviously a particular sensitive point in the immune response, right? That doesn't have redundancy. If we didn't have antiretroviral therapy, then we would eliminate everybody who wasn't CCR5 delta, right? So they would all survive. And our new population would be built from people who don't express CCR5. But there's there's nothing in each of us now that can survive the loss of CD4 cells. There's no redundancy that takes care of that problem, or people wouldn't die from AIDS. But maybe if there was population-wide infection, then there would be a few people who survived and they would have something, but it doesn't exist at the moment.
I've already been said, but can you transplant CDR or 4 cells? Like, because if that's what it's wiping out, why can't they either synthetically make it or dump more into your system?
Wasn't a Berlin patient a stem cell?
I mean, they need to be matured in the thymus, which is kind of hard to do, I guess, through transplantation, I'd imagine.
So they, mature CD4 T cells would be educated in somebody else's.
Right.
So,
Like,
so
you
they
take
would,
someone else's blood.
so you have graft versus host disease, basically, so.
But if they get a close enough match, like
Yes.
like if you have like a bone marrow donor or
Mhm.
something like that, it's the same type of.
Yep.
I think if I recall correctly, the Berlin patient, who's like the one confirmed case of total cure from 2011 was, he had stem cell transplant, and I think it might have been like stem cell lymphocytes that were already genetically resistant to the HIV virus. And what they think may have happened is that the transplanted lymphocytes were able to eliminate the infected ones, and essentially his or the patient's original lymphocytes that were being infected by HIV were wiped out, and the stem cell lymphocytes were able to proliferate significantly enough that they pretty much replaced those that the HIV had originally infected. So I guess in a way you could, but that's the only case of that, and again, that was just a couple years ago, so there's a lot of work to be done as far as finding more details about that.
So when we're talking about blood transfusions, when you get blood transfusions from another person, they're irradiated so that the white blood cells can't have a response against you because you're not HLA matched. When you get a blood transfusion, you're just marked for the ABO blood type.
But if like I had a twin sister, I have an identical twin sister, I got it, I could use her.
Mm-hmm. Eventually, until she ran out of blood.
You're right. Just you'll have another blood.
Just need a close one.
So, good job. I think you got all you can out of them, guys. I'll let you teach the HIV lecture too. So this was good. So we'll start again at 3. Anybody who has to take off? Chloe, take off. Good job, guys.
Thank you.
Thank you.
So first step, we already have a clone.
Yeah.
First,
We just get rid of the antiretroviral and just start over. It wouldn't do population. New population.
This year's right?
I have no allergies.
Busy Fridays.
I'm throwing this out there.
How was the
But
wedding?
It was really nice. It's kind of
HIV.
weird. Let's not jump in there, you know, people with cat allergies.
Yeah, my parents just 10 years ago. Neither of them were married, but it would be weird going 10 years.
Yeah.
That's what it was.
It's like, I mean, it was really nice. And I was like, this is a lot.
So like, actually, no, that's so, I had like, I thought,
Yeah. Oh, I didn't know that.
I was there, like, you're being,
Jeff, those people.
What?
I sound like that.
I've been a long day.
What?
Late night too long.
Yeah, I just want to check one thing.
Walkthrough.
I had just like this. Presentation.
You can try to do that.
Like I'll sort of see where I tried to get some work done on it.
Yeah,
Because I was like Tuesday, but I have
Get through the border.
So I can turn it Monday.
Like, it says return to Canada.
Still a bit of a late night.
It's not that hard to get
What?
I remember like in Smith, I would be like the one day I'd be up at like 3 in the morning.
I'm like a late night work
I can't
person.
work during the day.
I mean, I can, but I just prefer to stay up late and do it. That's gonna have to change now.
You want.
I always sign everything I do.
Yeah, that's in chemistry.
Oh, we got chocolate.
Why are we going to choose hall?
Not exciting.
Yeah, I know, really. I'll take a great nap. Getting arrived was cold or something. Me and Sappy. Bye, guys. I'll miss you too. See you Monday morning at 6.
Stop.
She's
Why is she leaving? Why is she leaving?
her flight.
From Texas.
So, haven't said it everyone else. Everyone's going to Texas.
Yeah.
What?
It's alright, I didn't go to Texas once.
No, I'm not going this year, so that's fine. I understand.
Texas, Texas.
Just kind of missed it, so got rid of it. Didn't watch it just now, actually.
No, I don't.
I don't watch it either.
guy I went to high school with plays for Memphis.
Grizzlies?
Yeah.
Wait, what? No way.
I'm not kidding. I had genetics class with him.
What's his name?
Tony Rowan.
This is my application.
For real.
Look him up.
Look him up.
Tony what?
WROTEN.
I'm worrying because there's like 14 jobs at stake here.
Good. I mean, he was pretty much a basketball star at my high school.
From the NBA.
He did that?
You guys doing sites?
I think starting the week after break, we just finished up. We just finished it.
Look how much
Stayed like 50 minutes extra on Wednesday. Finally got activated.
I like never
so
saw
fired
him
up.
live.
I don't think me and Abby only ever got, we didn't get, we got mini, we got like minis, but we never got.
There were so long
So now we go through like different.
We literally, we didn't get any until like 4:50.
He was drafted
on Wednesday. And we were just like, and we hadn't gotten any of the week before either. We were just so fired up, we finally got it. Nancy was sitting there for like 30 minutes trying to help us.
Is that frowned upon?
Awful.
I know.
Yeah.
I was like, I'm not leaving
I'm not
in
going
this unit without getting a single one. So I can't.
ACL injury.
Yeah, it only gets worse when you have to like record from it for
You don't deserve to say
30
that.
minutes.
You don't know.
It pops out all the time.
Suddenly all these basketball players were like, let's join the football team. So they did.
Yeah, we were like, quick, action, really quick to see the ball, and quick to take it off. Like,
Still like top 10.
I hardly think so.
I don't know.
You're leaving out like all the other
The other one is yours.
That's pretty insane.
Except for your mom.
You're essentially like
Good.
quarter of
You've always got her.
I never watched the basketball games in high school, so I should have. They were good, since they were good.
Probably the best.
Yeah. Yep. There was so much controversy, like every year there'd be something like, teachers give him a D so he can pass high school, or something like that, you know, something ridiculous.
D is the best.
Or like C or whatever, D or C, so he can pass, because they want him to go to play.
How did you have this classes in there? How did you get like the higher level classes?
He's 1 year ahead.
Oh.
Yep.
That makes sense.
Ride with Coach to the Portland Jet Card.
what?
He sent an email about riding with Coach to Portland Jetport.
You know, all three assistants are driving. Ready?
Yeah.
This is what I said, yeah, they're all, so one of them's just sitting in the back for the drive from here to Austin.
Bloom.
Bloom,
It's
it's definitely
Bloom. It's definitely
Bloom.
Bloom.
It's Bloom.
Oh, because I have to drive the
They have to drive the boats down.
Yeah,
That's
because
like the
Michaela
worst
and
job.
Seth have this like undeniable blonde, and
Why don't they
then
have
Bloom is
some
just kind of
cute
like, they're kind of, he's like shafted from the group. It's like,
And
oh.
they leave, they left at the beginning of this week.
They like take their time.
A little bit, yeah.
Yeah.
That's like, I feel like that's probably why all the assistant coaches leave after a year, which is the worst part.
I think it's probably kind of fun.
I guess it depends on the coaches.
Last year,
Like,
Jesse had to drive the trailer to Ontario, or wherever Hudson is.
Wherever his,
No, he drove to Hudson to get votes.
oh, that's right. That's over.
Where is Hudson?
It's in Canada, New London. I think it's in Ontario.
Ooh. Fun.
Did he drive down, did Jesse drive to Austin alone last year? Or did him and Devin?
I think it was Devin.
That would have been such an interesting car ride.
I bet Jesse wants to kill Devin.
He probably just had Devin sitting in the back.
Oh.
That's with the giant like maze
immune system.
I'm pretty sure
Definitely
Michaela
doing yoga in the bed of the truck when I drive down.
Should be Nerf pellets.
I
But what if there's
like
like
that
10
Michaela
of them? You just can't shoot them all at the same time.
That's why I always travel in packs. I always take the long route, no matter where I go.
Incredible.
When you're in Spain, did you like check out like a bike? Just like bike pass? No. Because like I bought like my yearly pass one and I thought like they cancel it like at the end of each year. But and like renew another year.
Of course, that's normal.
And like, and
well, I
I
guess
tried to
I'll
like
watch that.
cancel it, but I don't know like how. And there's the number that's been called. I cannot reach it by myself.
Oh, shit.
Oh.
I don't know.
Like on the floor one day, making fun of the other side, just like this side.
So we do. You'll never make it.
That's what I've been doing. I'd be told if there was
A zombie
any
apocalypse.
particular area of people that would get me.
Why would you want to do that? It's like
Track team. They almost got me last year.
Who?
You play that every year.
He's won every year.
Wait, you
Right.
won?
the NBA.
I think you like run around with the socks.
Yes, among other.
I can't believe I've never seen you. I've seen that like.
Yeah. Did you meet him?
I didn't see him.
He's always like his pants like always falling
I heard
down.
about it.
Oh, I messed up.
Yeah.
Well, last year.
Yeah, I
Last
met
year. Last year.
Freaking huge.
He was. I was
Like 7 feet
like,
tall.
I walked out the door and I see this huge guy and I was like, what?
He,
What's going on here?
whenever he got a kill, he would take the human's red wristband.
Because like the guy
and
who
sew it
was
together
like,
and wear it around his head.
Ten. I did see that actually.
Is that how, like,
This is one of his like new things.
So were you guys like big time enemies?
He came, he didn't come out to me once. Don't need to tell you that he didn't get me.
What are you guys talking about?
Humans versus zombies.
Humans versus zombies.
That kid got me
Apparently
last year
Cody
and I was
has
so mad.
Well, apparently
Yeah.
this kid's won.
Cody's
every
still
year.
wearing his
What?
bracelet.
Silverman won freshman year.
Yeah.
Because the whole like Friday night thing where you're supposed to go and everyone who's still alive, we had a regatta, so we left Friday morning. So Silverman was still alive.
If you don't play, can you really win? Like, I play the game.
Cody's like a.
Cody, it's just like a couple of weeks of you just being really nervous all the time.
Why do you do this?
Can't even talk to Cody.
Freshman year.
So everybody's divided into two?
Well, it
Starts
starts
with one.
everybody has a red
Except
wristband.
for who's
And
patient zero? Like
one person has a yellow wristband.
who? How do they determine who that is?
Whoever
You
And
don't
then
know.
they attack each other. And then the yellow
And then,
people
like-
try to attack the red people.
If
It's
the yellow person
zombies.
tags a person with a red wristband, then at the beginning of the game, should you become a zombie, you're issued a yellow wristband and you put it in your pocket or whatever.
So you don't even have to display it?
I think
No,
you
no.
You do, but
You're
you
not
have to
a
display your
zombie
red
first.
wristband. And then once you get tagged, you remove your red wristband and put on a yellow wristband and go tag other people with red wristbands. But then the plot is complicated by the fact that humans can kill a zombie with a sock or a
I thought you
Nerf
just
gun.
stunned them.
Well, of
Sock
course,
grenades get 5 seconds stun, I believe.
yeah, of course, for the gameplay, you don't actually kill a person, you stun a person. But in the story game, you're effectively killing a zombie. And so when you tag the zombie with a sock or a Nerf pellet, they can't be involved in the play for 15 minutes, and you can make your escape. Yeah, it's my favorite time of the day.
And is anywhere off limits?
Yes, academic buildings, commons, the library are all off limits. Commons and the library are always off limits. Academic buildings are only off limits 8 to 4 during school hours. And so I wanted to organize like a kind of slumber party in Pettingill where you stay and watch and that would be fun, I think.
Then your room is safe.
Wait, what?
Right, your room is safe. Your dorm room is safe.
For what?
For
Zombies.
zombies.
Oh.
Apparently Cody's are.
I'm so sad.
Cody is resident.
Really?
Yeah.
I remember, didn't you do that freshman year?
Yeah,
Holly?
this kid ambushed me.
I was feel terrified all the time.
Yeah.
I like
Cody,
sophomore year.
freshman year in Milliken, by day 2, there was
couldn't do
7
it.
zombies and then me,
Yeah.
including my roommate.
Yeah. Yeah. And then no one really did anything, so I just kind of said, your room is safe, not your dorm. So I just kind of didn't really do that much. I just came in the back door instead of the front door for a week, and then I actually tagged on like Friday.
I
That's all
got
I
the
did.
ambush coming out.
So it worked out pretty well. They were pretty lazy.
You know how the fire
really-
goes outside from the
Get tagged going to the bathroom.
You can't ever get complacent.
Oh, bathrooms are also safe.
What's the prize? Like, what do you get?
Yeah.
So
Like,
Cody's,
what is it?
Cody's alive and talking to him. That's the prize. You didn't beat me.
I remember doing that freshman year. I was so nervous. Like,
Yeah, definitely. I feel like I would just be terrified.
I was so embarrassed. I was confident.
But I'm so bad at math.
I one whole night. Yeah, I was like, I'm not listening to myself ever again.
You know what? I'm sorry.
I've never done that.
Like this when I was like coming back, and it's not though, I swear.
Nothing scary. I just wanted to have to admit it.
Like, she came to like, I had just been
I know.
So I just don't like when it has first like. Honestly,
I don't think I realized that like
it was like a public laugh. Why is everyone watching me come in?
Alex said I waited
I
inside
did,
it like
like,
I was hoping for all of us, but I just
Running towards the I'm like, my run, because like I did that like an idiot, and like it hurt my ankle more, and I was like, where he's
Like I did in my
she said
we had a belt created. We made this little belt, like a duct tape one, where you have all these other stuff, so you can put socks on it.
With chocolate, but if you have specific hits, like you have an assignment, you get as a server, whoever took out the most of their assignments during the week.
I mean, I guess it's better.
Draw corn.
Oh.
Yeah. She was like, you're like the only person I know. Last time, so
Yeah.
So we all have a did you apply?
I would, I could count, but
I think there's like part of the band where you
I see.
they like
Let's do it.
it's really not healthy for me, but.
Don't make Karen yell.
like so they can show what
Right?
And then, zero score.
You know, for the really
Yeah.
Got your emails.
Huh?
I said I got your
Just
email.
peanut butter and
Saying you received it.
Okay, here we go. So we like to hear. Polish off the weekend. Get you going on vacation or rowing or whatever. So if you don't have AIDS and you have T cells, it's not fun.
We know where you were going with that.
Really?
We're just so sleep deprived.
Little punchy. Okay, so this is what T cells do in health. And we'll talk about disease the rest of the semester. So you'll remember.
Oh my god.
Okay, well, this is going to be an interesting hour. That T cells are not so much in the mucosa and the dermis. They're predominantly concentrated in lymph nodes, and so for antigens of foreign antigens to get to T cells, they are trafficked there by antigen-presenting cells like dendritic cells. So when you step on the tack, the bacteria that are injected by the tack are picked up in the skin by dendritic cells, and the pathogen, the PAMPs associated with those pathogens activate the dendritic cell so they can migrate through the afferent lymphatics to a lymph node and display the antigens they picked up to T cells, the T cell areas of the lymph node. And so obviously there are lots of different kinds of antigen, foreign antigen, and they're processed in different ways by dendritic cells. So extracellular bacteria can be recognized by pattern recognition receptors like scavenger receptors and endocytosed, and then digested and presented on class 1. There's also a process called macropinocytosis, which is basically sipping of extracellular fluids by dendritic cells. So not a whole intact bacteria, but large molecules. The dendritic cells obviously they themselves be infected with viruses. And actually, dendritic cells are high expressors of CCR5. So dendritic cells are actually infected by HIV as well as T cells, as one example. And so in that case, when they're infected by viruses, they present on class 1. There's so-called cross presentation, in which extracellular antigens are picked up and escape into the cytoplasm, in which case they get presented on class 1 because they've gone to the proteasome and are secreted back into the endoplasmic reticulum. And then there's actually direct transfer of cytoplasm from one APC to another. So this is the conventional ways that we've talked about so far, where there's phagocytosis and presentation on class 2, but there's a number of ways that APCs can also present on class 1. Okay, so the dendritic cells are passing from the site of infection with their antigens. The T cells, we talked in an early lecture about how they continuously recirculate. So T cells are in the circulation, and they are constantly weaving through the high endothelial venules into the lymph node. And then if they don't recognize the antigen, their antigen in that lymph node, they'll pass out into the efferent lymphatics and continue passing through the lymphatics until they re-enter circulation. So there's two ways that the naive T cells can come into contact with their antigen, either directly out of the circulation or by draining from another part of the lymphatics. Okay, so we talked about the fact that this is a very similar picture to the rolling, tethering, and sorry, go ahead, Ryan. Just shout if I don't notice you.
Is for the slide about how the dendritic cells are able to come in contact with different viruses. So how is it that the virus is able to infect the dendritic cell without it being either taken up by macropinocytosis or the phagocytosis in general? How is the virus able to get in and infect the cell?
So just like you showed for HIV, it can bind a surface receptor and inject itself into the cell and actively infect.
But it ends up being the same response, essentially, right?
The
as
same.
far as the dendritic cell is still, for the most part, able to get the signal out to the lymphocytes.
Well, it's able to. So you guys still have a virology class. Is there still a virology? Yeah. So there's different types of infections. Some viruses don't kill the cell that they infect. Some immediately kill it. But so some dendritic cells get infected by some viruses and can still traffic to the lymph node. Some die where they are and get taken up by other antigen-presenting cells. So you get bits of dendritic cell and virus that get taken up. So
yeah, just because I was looking at stuff before, it was saying that a lot of times it's able to get to the lymph node, but not necessarily present properly, and then so the pieces are able to pass from dendritic cell to dendritic cell. But So it also can happen that they're not able to even travel to the lymph nodes and there's ion contact. And so is there a way for the signal to come out then if the dendritic cells are getting infected and then dying?
So then dead cells are phagocytosed by macrophages and other dendritic cells to be cleared. So dead cells are always getting cleared by macrophages.
And so the virus will still be taken up by those when they're...
Right, so it'll be presented on class 1 by the macrophage. Is there another hand? Okay, so remember neutrophils stay and monocytes stay in circulation until there's an active site of inflammation, and then the endothelium upregulates VCAM and other molecules to help recruit the cells of the innate immune system out of circulation. Lymphocytes are different in that they're always leaving the circulation, and so it's mediated by the interaction between LFA1 on the lymphocytes and ICAM1, as well as some other selectins of the high endothelial venules. Even without inflammation, there's constant recruitment of lymphocytes and their unique cell surface receptors out of circulation and into the lymph node. So that happens. It happens more often in inflamed tissues, but even when there's no inflammation, there's constant recruitment of lymphocytes out into the lymphatics. Okay, so I guess not surprisingly, that process that recruits lymphocytes out of the circulation, the interaction of LFA1 and ICAM1, is the same interaction that stabilizes the interaction of T cells with antigen-presenting cells. So APCs also have a lot of ICAM1. So in addition to the interaction between the T cell receptor and class 2 and CD4 in class 2, the interactions of LFA1 and ICAM1 help to stabilize the close proximity of those two cells. So when the two cells come close together, mediated by this adhesion, if there's a match in the TCR to the class 2, then you get activation and continuing cell contact. If there's not specificity of the T cell receptor for peptide, then that bond is broken and they move on to the next antigen-presenting cell. So, as we've talked about before, there's constant surveillance of what antigen-presenting cells are expressing by T cells in the lymph node. So they're always exposing themselves to the dendritic cells to see what peptides they have on the surface. Okay, so up to this point, we've just been talking about dendritic cells, but again, those aren't the only cells that present antigen. Macrophages and B cells also present antigens. And the ability of different cells to present antigens varies. So dendritic cells present a lot of viral antigens. Macrophages are particularly good at phagocytosis and intracellular killing of bacteria. So you see that macrophages take up their antigens by phagocytosis, whereas dendritic cells have macropinocytosis, phagocytosis, and tissue transfer. And then B cells obviously have the B cell receptor, so they have to have specificity through the B cell or the antigen, which they can take up and then present peptide antigens as well. And so this table just summarizes the fact that the different antigen-presenting cells have different proclivities for presenting different kinds of antigens, and they do it in different parts of the lymph node. But all the cells are there interacting with each other to increase the likelihood of a specific TCR coming in contact with its antigen from one of these antigen-presenting cells. Okay, so just like we talked about 3 signals of B cell activation, there are 3 signals of T cell activation. So the first is the recognition of the specific peptide MHC combination on an APC. So that that interaction that's stabilized by LFA1 and ICAM1. We talked about that. So when antigen-presenting cells, like dendritic cells, are exposed to foreign pathogens, those also activate other pattern recognition receptors on the resting antigen-presenting cells. So that causes the upregulation of co-stimulatory molecules like CD80 and CD40. So now the resting antigen-presenting cell not only has antigen, but it has additional adhesion molecules and signals on the T cell side from receptors for their activation molecules lead to the second signal. So there's a series of intracellular signaling events from the TCR activation and an additional signal from the co-receptors. And then the result of those first two signals results in the secretion of cytokines like IL-2 and IL-15, and those have an autocrine effect on the T cell. So they can affect other cells, so IL-2 can affect the antigen-presenting cells and B cells, as well as binding back to T cells. So basically, there's 3 steps the T cell has to climb over to activate and proliferate. It has to recognize its specific antigen, has to be interacting with an antigen-presenting cell that somehow has also recognized that it's been exposed to foreign antigen, and then it has to have enough signal from those two to produce cytokines to activate the T cell in the autocrine fashion. Does that all make sense?
What's the, right after the cytokines are released, autokine?
Autocrine? So it means you make it and it activates your cell. So it has receptors. It makes IL-2, but then it has receptors for IL-2. So it's a positive feedback loop, basically. You're releasing, and it's not just that one cell is making cytokine for itself. Basically, you have an area in the lymph node where one bacteria doesn't make an infection. So you've stepped on that tack. You have a bunch of bacteria in there. You have a bunch of activated dendritic cells that have trafficked to the lymph node. They're all making cytokine. You've exposed a number of T cells with specific receptors for antigens from that pathogen, and they're all in a very close proximity, making the activating cytokine. So the T cells are getting activated, the APCs are getting activated, and so there's quite a threshold to get an effective immune response. So there has to be enough of the pathogen and enough specificity in that local area to generate a significant cytokine concentration. Okay, so this is just one of those 3 signals, the specificity between the T cell receptor and the peptide MHC combination. And it just reminds us that the T cell receptor itself doesn't do signaling. It needs the other, all the chains of CD3 in order to signal, and as well as CD4. So basically, you have to assemble this multi-molecular receptor complex, and the interaction of the T cell receptor with class 2 and its peptide leads to phosphorylation, intracellular phosphorylation of chains of CD3, and then also activation of CD4. So this diagram just lists some of the essential intracellular kinases that are involved in that activation, and knocking out any one of these specific kinases has an inhibitory effect on T cell activation. Okay. So just like one bacteria doesn't cause an effective immune response, the interaction of one T cell receptor with one peptide on one MHC doesn't give enough stimulus to activate a T cell antigen-presenting cell interaction. You actually have to get a whole bunch of the TCRs interacting with a whole bunch of the MHCs, and that's stabilized by the adhesion molecules we were talking about. you don't have uniform distribution of those receptors across the APC and the T cell, when you're getting effective antigen presentation and an immune response, all of those specific interacting molecules get concentrated in the region of the T cell that's interacting with the APC. And so that's why it's actually called an immunological synapse. So just like neurons have a specific area where they exchange information, APCs and T cells form a concentrated region of signaling molecules during antigen presentation. And if you use agents to disrupt the organization of that synapse, you can prevent effective activation of T cells, like if you disrupt the cytoskeleton so that those molecules can't be concentrated in close proximity. Okay, so just like we took a closer look at sigma 1, this is just a slightly closer look at sigma 3, the autocrine IL-2 response. So the IL-2 receptor is actually formed by 3 different chains. So naive T cells have only 2 of those chains. Activated T cells express a 3rd chain. So part of T cell activation is actually synthesis of the 3rd chain of the IL-2 receptor. So you can have a high affinity interaction. And now you have IL-2 and IL-2 receptor alpha, the high affinity chain, being released at the same time. So therefore, you have the cell that's making IL-2 and the cell that is able to bind IL-2 in close proximity. So one of those activation thresholds to activate the T cell is the producing of a high affinity receptor for IL-2. Okay. So far we've been talking about every T cell activation, and for the rest of the class, we're going to start breaking up different types of T cell response. So IL-2 or IL-15 is involved in most T cell activation, but there's lots of other cytokines, and the types of the other cytokines that are made in that activating immune complex of cells in the lymph node determines what kind of T cells you get out of that specific TCR interaction. So you have a naive T cell that sees its antigen, and the cytokine environment in which it sees its peptide MHC antigen determines what kind of effector T cells it turns into. So the simplest view of that is the one that Ryan was talking about in terms of Th1 versus Th2. So in a context where you have a lot of IL-2 and interferon gamma, you get a Th1 cell, and they make a lot of interferon gamma, leads to macrophage activation, B cell activation, production of opsonizing antibodies. Alternatively, a Th2 cell results from exposure to IL-4. They also produce IL-4 and IL-5, and this leads to class switching and antibody production. So that's the simplest view of the divergence of different effector T cell types. So slightly less simple is that we also talked about the fact that there are regulatory T cells that suppress the activity of. So you can imagine that when you are responding to a foreign antigen, you want to develop the ability to suppress your immune response when you've eliminated the infection. So the exposure to TGF-beta in that immune response leads to T regulatory cells. So in every immune response, you get some amount of effector T cells of all of these types, but it's the cytokine environment of that individual germinal center that determines the relative proportion of the different types of effector T cells you get out. Okay, so we have simplest, less simple, and we have slightly realer. So there's not just T regulatory cells, Th1 cells, and Th2 cells. There's also Th17 cells. So these are cells that make interleukin 17 and TNF, and these cytokines feed back on the innate immune system. So these are activators of neutrophils, and they're very important for immunity at the mucosa, like in the GI tract and in the lung. So now you can start to see why we have so much immunocompromise in HIV if we eliminate the T cells. We can't do cell-mediated cytotoxicity. We can't promote the maximum activity of the innate immune cells neutrophils. We can't have optimal antibody responses by making Th2 cells to promote class switching, which is necessary for IgE against parasite in addition to allergy. And we can't make T regulatory cells, which are important for regulating the immune system. So which of these things happens depends on in part on the environmental gamish of what sorts of molecules are in the pathogen that we're responding to, and that the resulting cytokine cocktail that the T cells and antigen presenting cells create in making that immune response. So like many other things, the type of immune response we make depends on the environment, our genetic makeup. Some of us tend to make more Th1 responses. Some of us tend to make more Th2 responses. So some of us tend to be more allergic, some of us tend not to be. And then it depends on our hygiene. What pathogens have we been exposed to? So some types of pathogens lead to more type 1 responses. Some lead to more type 2 responses. So, relatively clear? Okay, so we've done 4 steps now. Simplest, less simple, almost real, and the realest understanding we have so far is that there aren't just T regulatory cells, Th1 cells, Th2 cells, and Th17 cells. There are also Th9 cells and Th22 cells. And they're also named after Th1. So 1 and 2 are easy because we thought, well, there's 2 types of T cells, we'll name them 1 and 2. After those 2, the other ones are named for the predominant interleukin they make. So Th9 cells make IL-9, which is important for mucus production. Th17 cells make a lot of IL-17, which is important for extracellular pathogens like fungus. And then Th22 makes IL-22, which is important especially for the GI tract maintenance. So I'll just stop and say we don't need to memorize this. Just keep in mind that T cells are central to the immune response, and the type of immunity they mediate is affected by the cytokines they're exposed to during that immune response, and results in different functions and different products. And the downstream interleukin products affect the kind of immunity they mediate. And they're not totally fixed. They can, during proliferation and activation, they can be changed from one type to another. Nancy, smile. Okay.
You don't know how often I get that from professors.
Sorry?
I get that pretty often from
Oh, yeah?
professors.
Okay. You look like you're in pain. Okay. Excuse me, sir, my brain is full. Okay. We're almost there. We're almost there. Okay. So there are a lot of different kinds of T cells. Mediate all kinds of different immune responses. The cytokine environment of that germinal center determines what kind of T cells they differentiate into. So cytokines are only one of the ways in which T cells can be effectors of the immune response. Another way for CD8 positive T cells is direct killing. So they can secrete cytokines that kill cells. They can express molecules on their surface that bind to apoptotic receptors on cells. And they can release contents of granules directly onto those cells, kill them. So T cells produce cytokines that are inhibitory to cells that are virally infected, and proteases that directly kill, as well as mediating receptor-mediated killing signals. And then CD4 cells can secrete cytokines directly to mediate the effects of other immune cells, as well as recruiting other aspects of the immune system. Th2 cells, their cytokines and molecular surface molecules are essential for the maturation of antibody-producing cells. So, the effector cytokines of Th1 are interferon gamma, which makes cells more virally resistant, GM-CSF, which recruits macrophages and neutrophils, TNF, which causes localized inflammation and has all those effects on the endothelium, whereas most of the cytokines made by Th2 cells target B cell maturation and the production of regulatory cells. Okay, so the Th1 effects help to promote intracellular killing. So macrophages that are exposed to Th1 cells and Th1 cytokines like interferon gamma are better at killing intracellular bacteria. And there's activation of co-receptors between the T cell as well as soluble products to be able to do that. But it's not only interferon gamma that helps promote intracellular killing. They also make ligands for apoptotic receptors. They make IL-2, as well as cytokines that promote proliferation of macrophages, as well as cytokines like TNF that help promote the recruitment of more white blood cells. And they can make chemokines to promote the recruitment of more leukocytes to the site of infection. That's basically a repeat of the same. Okay, so basically just digesting those specific T cell effector functions. So Th2 express CD40 ligand, and that's an essential co-receptor. So this is one of the 3 signals of B cell activation. So T cells need 3 signals, and B cells need 3 signals. The signaling of CD40 is one of the essential ones for B cell activation and proliferation. And then cytokine products of Th2 cells like IL-6 and IL-4 promote B cell maturation and differentiation into plasma cells. So remember that naive B cells just have BCR on the surface, and they have to go through activation and proliferation to become antibody-secreting cells. Okay, so not only do Th2 cells mediate proliferation and activation of B cells, but many other cells of the immune system can respond to Th2 cytokines. They make antigen-presenting cells actually produce less inflammatory cytokines and more, but they become better at phagocytosis. They increase mast cell proliferation, so this is why Th2 responses lead to allergic responses. They activate specific cells of the innate immune system like eosinophils, and they make antigen-presenting cells better at presenting antigens. So they take cells of the innate immune system and make them less likely to have a Th1 response and more likely to promote antibody responses. Okay. So Th1, Th2, CD8, that's the essential. This is the current, or more current, in terms of the effects of IL-17 and IL-22. So Th1 and Th2 cells predominantly affect other white blood cells, other cells of the immune system, whereas IL-17 and IL-22 actually bind on other cell types like epithelial cells. IL-22 promotes the health of epithelial cells. IL-17 causes epithelial cells to release chemokines that recruit neutrophils. So these are very important for mucosal immunity. So IL-22 remains mucosal integrity. IL-17 helps infected mucosa to recruit cells of the innate immune system. Okay. So we'll just finish off with mechanisms that CD8 cells use to kill targets. So two types of T cells, CD8 positive, CD4 positive. CD8 positive are interacting with class 1, so they're dealing with virally infected cells. So this cytotoxic T cell has recognized a foreign peptide associated with class 1. On this, looks like an epithelial cell. So it does its thing and kills that infected epithelial cell, and then has the ability to move on and continue to kill multiple virally infected cells. So it's not like a neutrophil that just explodes upon activation and is killed. T cells can continue to kill multiple target cells. So just like T helper cells or CD4 positive cells, activated naive cytotoxic T cells have to receive 3 signals to undergo proliferation and activation. And they produce their own cocktail of cytokines that leads to effector cytotoxic T cells. So the early ones don't have granules full of proteases and reactive oxygen species. Once they've gone through this activation and proliferation, they're full of vesicles that are full of those cytotoxic agents. So then when they interact with a virally infected target cell, they can release those granules to basically directly digest the target cell. They also release cytokines like TNF that can kill the target cells. And like I mentioned before, they have surface molecules like Fas and Fas ligand that deliver apoptotic signals to the target cells. So this is just a recapitulation of what I was talking about in the last slide, where T cells can deliver cytotoxic signals through membrane-bound molecules, proteases, and through secreted cytokines. Okay. So an aspect that we talked about for B cells is that when you have an affected B cell response, when a B cell has seen its antigen, it's gone through proliferation and differentiation, not all of the cells become plasma cells that secrete antibodies. Some of them become memory cells. So the idea that you can recognize the same antigen again. And the same is true for T cells. T cell maturation and proliferation leads to memory, to effector memory cells as well. So after that first activation and production of effector cells, I think in this depiction it's cytotoxic T cells, you produce some long-lived effector cells. And that may or may not be cytokine dependent, but they are around. There are more of them around if you get infected with the same agent again. And this is why the best vaccines are ones that stimulate T and B cell memory, because you're getting production of memory cells against different antigens of the pathogen in both the B and T cell types. And if you take blood from somebody who's undergone an immune response, you can tell which cells are memory and which cells are naive by the pattern of cell surface markers. So not important to memorize, but you can basically break down T cells by, you can identify T cells as being T cells because of CD2 or CD3. You can differentiate them as T helper cells or cytotoxic T cells by CD4 and CD8. And then you can use these other molecules to say, is this a naive T cell that's never been through an immune response, or is this a T cell that's producing, is a memory cell that's already gone through an immune response in the past? So one of the reasons that you can distinguish between naive cells and memory cells is because of those cell surface molecules. And some of those cell surface molecules are adhesion molecules. So now, whereas your naive cells undergo that constant recirculation through the high endothelial venules and through the lymphatics, the altered expression of adhesion molecules now on a memory cell allows it to redistribute into tissues from its primary immune response. So a T cell that had its primary immune response in the gut or in the brain or in the lung, the memory cells from that immune response are more likely to redistribute to that tissue. So not only do you have more cells specific for antigen, they're more likely to reside in the target tissue of where the naive T cell had its response. Does that make sense? And that's all regulated by different cell surface molecule expression and the affinity of those cell surface molecules for the particular tissue. There you go. Okay. So, and this just points out the fact that we started by saying that dendritic cells bring antigens to the lymph node, and that's where most naive T cells see antigen for the first time. However, effector T lymphocytes with those new different surface molecules redistribute, and now those effector T cells can encounter antigen in the tissues themselves. They don't have to wait for antigen to be brought to them in the lymph node. They're out there in the particular tissue where they had their immune response, and now they're actually surveying whatever the tissue is for more of that antigen. If they don't find it, then they die off, or just continue to circulate as memory cells. But they basically are looking for more of the antigen from that naive response. So not only in memory, not only do you have more cells specific for that response, but the activation threshold is lower. So for that naive immune response, remember we have three signals: the T cell receptor signal, the co-stimulation signal, and the IL-2 signal. The memory T cells have more of all of those molecules, so you don't have to be exposed to so much antigen in a memory response as you do in a naive response, and that makes sense. You want the threshold to be lower. You want it to be really hard to kick off the immune response the first time, but once you know that you have been infected, if you get infected again, you want to kick off your immune response with a much lower threshold. So this is basically everything we talked about today. The activation of naive CD4 and CD8 cells in the lymph nodes responding to professional antigen-presenting cells, receiving three signals to proliferate and activate. And then the results of that proliferation of T cells are effector and memory cells. Effector cells for cytotoxic T cells can pass out into the tissue and look for virally infected cells, which they kill with soluble molecules as well as receptor cell-associated molecules. T helper cells, depending on your genetics, the environment, and the cytokine milieu of the lymph node, develop into at least 2, if not 5, different effector types that can then leave the lymph node and mediate their various types of immune response, like B cell maturation or enhanced intracellular pathogen killing. Questions? No? Saturated? Ready to go on break? Good. Okay. We're done. So I'll make every effort to have your tests back to you by the next time we meet, which is 2 weeks from today. Huh?
You don't have to.
Well, otherwise, it's not going to do anything. Getting ready for the next one.
I might see you later.
What? Yeah. Oh, I'm gonna leave tonight. But have a good break, although I guess